Okay. Good morning, everyone, and good morning, Dr. Reddy. Thank you so much for the invitation, and I feel honored to be with you all this morning and to share some of the my views about the bioelectrochemical systems and share my experiences and maybe some of our research. And engage in discussions, hopefully. And I'd like to draw some attention after the meeting as well. If you have any interest in learning any of this more or looking for opportunities, please get in touch with me. So as Dr. Reddy, again, thank you for the generous introduction. And so as the title says, my talk will be on bioelectrochemical systems for circular economy and environmental sustainability. So would like to start with acknowledging all the funders and collaborators and the institutions, and most importantly, my own research students. Without their support, all of this would not be possible. So big thanks to my students. Let's look at this outline first. I will like to give you some introduction and some basics of what is bioelectrochemical system. And then we go from there. We try to understand the context of bioelectrochemical systems and from the point of view of wastewater and organic resources, their treatment and management. And then we'll talk about circular economy and again, the role of bioelectrochemical systems. And followed by, we'll look into energy and environmental sustainability, or where we are, especially with this, the whole range of technologies that we have under bioelectrochemical systems. And then we'll finally close with the challenges and opportunities. I would like to first give a brief introduction about what it is exactly. So if you look at the definition, a bioelectrochemical system is a technology that integrates microorganisms with electrochemical components. So these electrochemical components, meaning that the chemicals could be the food, the microorganisms that they need, and obviously electrochemical. So electro compounds, meaning that those are electrodes, and of course we also have other elements I'll discuss with you. So simply put together, it's a microorganism and an electrode interacting with each other to provide or to derive some useful products. In this case, we are trying to convert organic waste into electricity or valuable products such as methane, hydrogen, and those are the high value energy products. And there are other high value chemicals as well. Now, the role of microorganisms sometimes is interpreted as biocatalysts. That means they allow for the transformation of chemicals from one form to the other. So in this case, the role of microorganisms is highly pronounced, and it would not be possible, that kind of reaction would not be possible without the mediation of these living organisms. Now, bioelectrochemical systems, when we think about this word, I think a lot of you might be thinking about microbial fuel cells, right? So, and then maybe you're thinking about microbial electrosynthesis cells. And the other commonly utilized or explored configuration is microbial desalination cell. So these are all three different technologies. They have three different functions. One of them, the first one, microbial fuel cells, is about generating electricity directly from the substrates. So substrates here means wastewater or any other organic resource that comes from any industrial or agricultural wastewater sources. And that potentially serves as a source of energy or food for microorganisms. Then we're talking about microbial electrosynthesis cells, where microorganisms actively convert or contribute to the process of delivering high-value products, such as in this case, I'm showing hydrogen as an example. And then we do have microbial desalination cells. and microbial desalination cells combine or integrate an ionic separation process, and they also integrate one middle chamber that is separated by anion and cation exchange membranes. That is to selectively drive the ionic migration between anode and the central compartment and cathode and the central compartment. So depending on the ionic imbalance, that would be the driving force for this migration. And that process is supposed to achieve the desalination effect. It's supposed to provide that. And then so we also get additional benefit of cleaning more water with what we are already doing in a microbial fuel cell setup. So I will be going through a little more detail into microbial fuel cells today, only because it's a huge topic. And as you will see in the coming slides, there is a lot to talk about, but we do not have time. But also at the same time, I want us all to, kind of advance to a level where we are today in the when it comes to the state of the art of research and the industry where they are, and so what needs to be done. And so the focus later on would be, of course, after I introduce you a little more about bioelectrochemical systems, and then we will narrow it down to microbial fuel cells. So other technologies, or you may have heard of these, plant microbial fuel cells, where you would use plants in addition to microorganisms to do what we want them to do. For example, in this case, of course, sun is very important for plants, and then they grow, right? They fix carbon dioxide, they absorb, and then produce the molecules that are required for growth, and so on. And then you will see that some of the molecules of those are being actually excreted through roots, and then those will be present in the soil. And of course, that's where we call them plant exudates. There could be different types of substances. Here we're trying to show as glucose as a simple representative molecule. So if we were to imagine a microbial fuel cell that is integrated with plants, then that would be that where you will have anode integrated into soil, and then you would have cathode exposed to air. Air is usually used as the cathode most of the times. Now, enzymatic fuel cells, which we do not see much in environmental applications, but they were earlier designs, but they're still being explored actively for various other purposes. But this is going into more into biochemical point of view. And so this does not necessarily require the whole cell, but this is an enzyme you can do this. Then we can talk about sediment microbial fuel cells, which is another important application or the technology that would be interested in, where we want to, bioremediate the pollutants and contaminants that are stuck in these sediments underwater bodies. So if you look at bioelectrochemical systems, they are not a really as novel as we would think. They've been existing for a long time, and the very first effect of something like an interaction between a living species and metals and electricity, anything like that, was already invented in 1786. And then, of course, we haven't seen much documentation after that. The later documentation we would see is in 1911, Potter. That's when he pioneered some of the biological components, integrating them into electrochemical processes. And then 1950s, microbial fuel cells emerged as a pioneering systems because of the interest in different directions. Well, this is to be noted that initially all of the research was, conducted by those researchers who are really interested in understanding fundamentals. So their vision was not really probably for practical application that we are thinking of today, but they were initially attracted by those fundamental researchers who are trying to understand this basics. And then 70s, the enzymes came into place. In the 90s, and we started talking about nanotechnology and then material modification with all the technology and science advances that we had. And then in 2000s, the technology has a different configuration, different forms of bioelectrochemical systems emerge because of the environmental emphasis across the global community. And then today we will see that our focus is on developing sustainable systems. So that means we need to make sure they are cost effective, they are socially acceptable, they're environmentally friendly, and so on. And so those are actually higher challenges that we're facing today, and then we're trying to march towards in achieving even circular economies. So those are different things that we want to talk about today. So I would also like to give you a broad view of bioelectrochemical systems before we move on to the microbial fuel cell concept. Yeah, bioelectrochemistry is the basis for everything. It's a big discipline, large discipline, where we would see microbial electrochemistry, enzyme electrochemistry, and then DNA protein-based electrochemistry taking place. So if you're wondering, more of DNA protein-based and enzyme-based chemistry is more used in medical applications. So in that, the subset of disciplines is the microbial electrochemical technologies. So all of these technologies that I just discussed with you, they all fit in this microbial electrochemical technologies category. And in that, we do have what is known as primary MET, microbial electrochemical technology. And then there is secondary meaning that two processes are integrated in which one does not necessarily have, or the MET is the secondary part that is indirectly utilized to achieve an effect. For example, to maintain a pH balance or to draw solutions from one compartment to the other using a membrane or something like that. They're called secondary METs. But wherever we do use microbial technology, sole configuration with the intent of getting either electricity or high value products, then we call it primary microbial electrochemical technology. Now, this is a big word, right? It's a mouthful of words. As we see here, you can also see different variations of the same thing. If depending on the focus, for example, microbial electrocatalysis is looking at the microbes as biocatalysts. Okay, so if the focus is looking at them as participating in something, or microbial electrosynthesis or microbial electrochemistry in general is between microbes and electrodes. Okay, any interaction between microbes and electrodes and in the chemical environment, right? It's all microbial electrochemistry. And so again, the focus, if it is synthesizing something, then you would call it microbial electrosynthesis and so on. Let's move on from here. So coming to microbial fuel cells, you will see that there is anode and cathode. Okay, so anode is on my left side, and then cathode is on my right side. And so anode is where you have the biofilm, and so it's all of the time is biotic process. And cathode can be biological process, but it's a lot of research focuses on abiotic cathodes, meaning that you only have electrode, you do not have microorganisms or biofilm working actively on it. But whereas in anode, we call it bioanode because it's almost all of the cases we have microorganisms forming biofilms. And then, of course, there are also biofilms in the water suspender. And so they're all actively working. And then when they are actively producing electrons in that specific groups of microorganisms are called electroactive microorganisms. And then if it is a biofilm, then we call it electroactive biofilm. So what they do is they take these organic substances and convert them that through their metabolism into energy and other stable products, I should say. So that would be one of them is carbon dioxide. That's the most stable carbon compound that we have. And then electrons that are released. So you will see that if you see, and as you notice here, the microorganisms are attracted towards this electrode because they need that electrode to be able to respire and release that electron, which will be taken by the, in this case, electrodes acts like an electron acceptor. So, but for the challenge for the microorganisms is to find that nature, find that place where they're able to attach themselves to the microorganism, to the electrode, and then release the electron, and then which would flow through the current electric current loop, right, circuit to reach cathode. And so this flow of electrons is electricity. That's what we know. And then we need an electron acceptor. So we have donors, electron donors, that is the substrates for for wastewater simply is the electron donor. So in this case, if you see at anode, we with the simple representation of acetate ion that is oxidizing to release electrons here in bicarbonates. And then at cathode, we will see that oxygen from atmosphere is being reduced. So there is oxidation going on in the anode, and then there is reduction going on the cathode, and that completes the reaction cycle. In that process, we see the flow of electrons going from the anode to the cathode, and that generates electricity. Now, the electromotive force is very important, expressed in terms of voltage. And then you'll see that these are against standard hydrogen electrode. I'm not going to go through a lot of details here. There's a lot of thermodynamics that to be understood. But what I would like to say is that you want to match these anodic reactions where you have the substrates that are being converted into stable products and electrons. And then we do have the other compounds that serve as terminal electron acceptors, so such as oxygen in the cathode chamber. So we need to match them so that the more the negative and the positive values that are associated with the anode and cathode in terms of electromotive force, the higher the electricity generation potential for the configuration. So now from there, we'll see that there is a lot of possibilities. Again, we are looking at glucose, glycerol, malate, and sulfur as kind of model compounds here. But wastewater, as you know, it is a very complex resource, and where you have large molecules because you have proteins, fats, and oils, and everything, and of course glucose and everything in the different forms of glucose. So its molecular weight would be much larger, and it should be very much difficult for these microorganisms to degrade or break into smaller compounds. And so that needs to be taken into account. When it comes to cathode, you'll see that there is, again, a plethora of possibilities where you could use oxygen or you could use nitrogen-derived compounds that would also accept electrons, okay? And then you would have other metals and metal oxides that could also serve as the terminal electron acceptors. So as we noticed here, there is nitrogen-related compounds, and there is others. And of course, oxygen is the main player. So that actually motivated us to, because when we are utilizing oxygen as terminal electron acceptor to increase the cathodic reaction, which is a limiting factor, many studies, earlier studies utilized platinum catalysts, so which are very expensive. They were utilizing catalysts on electrodes to enhance that process, reduction reaction, but that is not acceptable because it's going to increase costs, and then it's also not environmental friendly. And so Our interest was in looking into biocathodes, and that was my area of expertise for over 10 years or so. We've been working, and we were the ones to present some of the ideas first in the research community. And so one of them would be a microbial desalination cell, which is having an in situ generation of oxygen produced by microalgae. And then when we looked at the system and it's operating, we could see the bubbles coming out of, popping out of this tiny algae cells. We were so excited that, we could see that it's an enlarged picture. It was amazing to just notice as you were observing the system work. And so that was one of our contributions, earlier contributions. And then later, again, in the area of biocathodes, if that means we're using again, microorganisms to do more efficient work there in terms of providing oxygen or in terms of converting nitrogenous compounds, right? So just simply removing organics in the anode is not sufficient to say that we're done with wastewater treatment, but we also have to remove nitrogenous compounds, and then that is where the regulatory standards are. And so our interest was to explore the possibility of integrating Anammox bacteria in the cathode chamber, so biocathode chamber. That's also again one of the first studies that we are also working on a patent. Some of these are patentable technologies. Now, with that big background of bioelectrochemical systems and narrowing down to microbial fuel cells, I would like to talk about the linear versus circular economy just a bit, so we will also see it in the context of wastewater management. So linear versus circular systems that we know of today. So linear economy is reliant on make, use, and waste, right? Or dispose. So that is what we know about for a long time. Of course, with education and outreach and the living standards going higher, I think people are now more conscious about recycling and reusing. That is already going on for, it's nothing new to us. But what it means to enable circular economy is that you are trying to keep these resources that you have already utilized, or that means the products in the loop. As you see the circle that represents the loop here, as long as possible, so that you are reducing the intake of fresh or virgin raw materials that we liberally extract from nature beyond its capacity, right? Sustaining capacity. So at some point it becomes unsustainable. That's why circular economy, what it means is when we keep these whatever we're talking about, these chemical compounds and products that in the loop, we are avoiding extraction of new resources. That means we are extending the longevity of resources, as well as we are cutting down costs. So that is contributing to environmental sustainability as well as the economy. Now, in wastewater context, we will see that there are different sources of wastewater: domestic, agricultural, and residential, and municipal, and then industrial, of course. So we need enabling technologies, right? So those are called sustainable technologies that could allow for circular economy to be carried out. And then the products that could be of interest to us could be nutrients, clean water, clean energy, and so on, other valuable products. And someone raised a hand. Please bear with me. We will get to you at the end of the talk. So when we look at the wastewater treatment, the philosophical way of looking at it, all we knew is, or the industry was always looking at removing it, getting rid of it. It thought waste is never seen as a resource. And so we had removal technologies very well developed all this while, or all our technologies are actually motivated or designed with the motivation to just get rid of things. We really don't worry about where they're going. But The new thinking is that we want the resources to be recovered. So I like to use the term used water rather than wastewater because the water is water, no matter what type of water it is, what the quality is. But it is going to be more respectful to say it's used water, and because after all, it's us who use this water, and but it is still full of resources. It's not really a waste source here. And so if you recognize that there is water, there is nutrients, there is other metals, valuable chemicals, and then that kind of presents as a resource. Now, we could use all of these bioelectrochemical systems that we discussed to recover and convert some of these organic waste into valuable products. But what about the thermal energy? That's one of the areas that I'm actually interested in because of my backgrounds. We almost never focus on thermal energy in this context. But wastewater does have significant thermal energy, although it is very, very low quality energy source. But that's something that we need to be thinking about because that's the majority of the energy that is embedded in wastewater is thermal energy. Next is only the chemical energy, and which we are after, right? So if you look at the thermal energy, the technology that I can think of is the microbial thermoelectric cells that would lead to a brand new discipline. It could be itself and an area, and that is something almost inexistent. It's not explored at all. If any of you are interested, yeah, take a look at that. Now, resource recovery from organic sources. So the reason I'm putting this slide here in the context of what we're talking is that we will see the application of anaerobic digestion from organic resource management perspective. So all of this organic waste that we have from coming from different sources could be subjected to anaerobic digestion, and then we can produce biogas. And of course, there are various products that can be, so the key will tell you what's the feedstock and then what kind of technology we would use. There's a lot of thermochemical technologies and then physical technologies as well, and then what kind of resources we can. I think we can extract, and then we are familiar with all of these, so biogas, biochar, bio oils, and everything. So let's come back to this wastewater circular economy. I would like to give you some basics about this and how it fits really. And then sometimes we do not realize that we are actually doing this already by working in this research domain, okay? So, and then I will give you an example of our own research. I cannot go too much into details. I try not to go because we do not, each of the study, I could talk for hours actually. So there is no way we can go into details, but please feel free to reach out to me if you need more information. Now, I would like to compare with what we do today versus what is actually possible, that is circular economy potential. So if you look at the carbon and nitrogen and phosphorus present in the wastewater, okay, if we're looking at that, we put it in terms of, a unit. That would be more convenient for us to understand based on a basis, right? A standard. So 1 kilogram of carbon and 1 kilogram of nitrogen and phosphorus. Suppose if we have to remove 1 kilogram of carbon, we will have to utilize 1.48 kilowatt hours of electricity. So this is electricity. But whereas if we put that carbon to work, it is possible for us to recover, in addition to saving this energy of 1.48 kilowatt hours, we would be able to, this number would be very promising for us to see that it is, so we're able to generate more electricity by putting them through beneficial processes. For the same thing, if you look at nitrogen, to produce 1 kilogram of nitrogen through the Haber process, right? It will take 13.44 kilowatt hours, but if we put it in processes where we recover nitrogen, that could actually bring an energy benefit. This is only looking at the energy point of view, but now you could add a cost to it, but that's a different story we can talk about later. So again, phosphorus. removal versus removal is very expensive, but if you're going to put it to or recover simply, that is actually going to save a lot of electricity. So now what do we get out of this wastewater? Organic soil conditioner with fertilizer, right? Even if you're selling it very cheaply for cubic meter, $0.33 $0.03. And methane, you can get nitrogen. You see that that's, those are very conservative values. But as you see, water is the most valuable resource that we see that has the maximum price tag that's $0.33 dollars for cubic meter. And that means that's just it's clear to us if when we are treating wastewater through these processes, we are benefiting, and then the most important resource that we are recovering is the water. Now, we'll look at the nutrient dilemma because I'm going to talk about the recovery of nutrients, and then we'll talk about energy, and then we'll close it. So in nutrients, we look at it over the past few decades, we have increased the demands or utilization by 8-folds and 3-folds for nitrogen, phosphorus. Okay, that is actually contributing to a lot of environmental challenges that we're facing today. And so the solution for it would be to recover as much as possible and put them back into the system again. So there is a huge disconnect. My research is driven by this background in theme that would look at in the disconnect or the sources versus sinks, right? Sources are agricultural communities and rural communities, but sinks are urban communities, right? And so there is a big disconnect. You have a source of production, food production. The food goes into urban communities, and then the waste is generated and discharged into the environment. So you have environmental impact all over because of the transportation of the products. And so we see that microbial electrochemical technology is playing a key role in advancing and our circular economy goals and then addressing environmental sustainability challenges. So as an example, we will say that, let's say if you have an agricultural industry where production is taking place and you have concentrated or simply domestic wastewater, that's what I was going to show you, and agricultural wastewater, such as like dairy farms and so on, we could extract those nutrients from the wastewater and then put them back into the fields, the crops. So that we're trying to look for a shortest route because this could be a lengthy process as well as you discover and then try to transport and then put that in the system. If it is in liquid form, it's going to be very difficult for us. And so it would be wise for us to recover these nutrients in the form of solids. So what we did is, because these materials have to be sustainable, if you want to blend something with soil, right, they have to be biodegradable or biologically compatible, right, biocompatible. So we used terracotta as a part, and then we also used biochar, which is a carbon, basically to replace electrode. Our terracotta material is working as a membrane, natural membranes for separating the water and the anode and the cathode. And then we do have wastewater in the pot, and so where we are trying to recover. We used even bentonite, natural clay material to to keep the things together and treat the wastewater in there. So this would be the final step of a process where you connect with the electric circuit, where you have the different materials. This is natural earth materials, right? And that we use for treating the wastewater in the different units here. What we have seen is that our process was able to the voltage potential and everything. It was very close and comparable to what has been reported in the literature. And of course, we have done a lot of electrochemical research in this case, and there's extensive study we have done. We were looking at what kind of resistance that we have to put in between the anode and cathode so that as the electrons flow through, the resistor, there is the electricity that's generated. And so those are the studies, and we looked at how much of the COD is being removed and how many hours it is taking. and what is the power density that could be achieved through the system. And we also looked at the trend or the kinetics of what's the time frame to achieve about 70% COD removal versus 90% COD removal. So actually, uh, within the first 50 hours, we were able to see the majority of the COD removed, and then the next 50 hours is actually only taking out 20% or 10 to 20% of the waste in the COD. That means that what it indicates is that when we are designing systems, there is some sort of trade-off, how much COD you want to remove and how much electricity you want to generate. Obviously, we don't want to work on the curve, this falling curve, or which will translate to the curve where it is the electricity production is slowing down. And so in real applications, we might want to just operate at this flat line where we are producing the maximum power. And so that means we will have to push water through the system, and that indicates that we will have to sacrifice the additional 10-20% of the removal capacity, which needs to be taken care of in another compartment or another unit. But I would suggest to utilize a biocathode. So that's where, again, biocathodes come into play. So now what next? Because I want to talk to you about circular economy context, right? So we have recovered all of these nutrients through these parts studies that we have done. And so we have done this. We studied everything. Everything is looking good. So next what we did is we crushed these materials, right? And this is my graduate student who worked on this project. And we mended soils. We took the straight soil for plants studies. And we, I think it was 1:9 ratio we used. And then we mixed it up, and then we have done these studies. And then you will see that the straight soil, which is the control, would have very less phosphorus, compared to our wastewater, dairy wastewater and synthetic wastewater versus, the control. And so nitrogen is the same thing as well. We haven't shown it here. For example, you see potassium. Again, some of these elements are not, very well available in abundance in this system. So that was really interesting to us. These studies were done at a facility at Mississippi State University with our collaborator in plant sciences department over there. And so we have done multiple studies in winter conditions and summer conditions, and also simulating drought conditions, and results were amazing, and they were very promising. So we will need to explore further to understand, because of course we haven't tested them through the full growth cycle. So these are corn plants, but we only tested them a maximum of 40 days, but they could grow up to like 3 months. And so there's more to study. So what we have observed is that within the soil amendments, these are the treatments that we are interested in, right? Because biochar and terracotta are also some sort of control setting, right? Against which we are comparing our soil amendments with the nutrients that were recovered from these wastewaters, right? So we saw that the synthetic wastewater actually produced much higher plant growth, including the height and areas and weight and everything. So that shows that this study demonstrates that it's possible to recover nutrients efficiently within, the with minimum number of steps involved in it. So that's a point here, and of course more sustainable by utilizing the terracotta and biochar materials. So there is no membranes, and there is no electrode materials that would be like graphite and things like that. And there is no catalyst that we utilize. So it's all environmentally friendly. And so now we see our research efforts are into membrane performances. We're looking at different types of membranes. And then we have hydroponic nutrient management, where we're trying to adjust the quality and recover some of the phosphate and address some of the quality issues, and electrochemical treatment, microalgae based analysis. These are all, again, we have limited time. But one more thing I would like to spend some time on is the energy aspect of bioelectrochemical systems, more specifically for microbial fuel cells. Okay, now before we go into any detail, I would like to say is that we cannot make these systems operate alone, stand alone. They need to be integrated, at least for now, because of their suitability. So this is one of our studies. I'm going to share the link with you. You can access this paper for more details. It talks about, COD and nitrogen generated per person per day, 110 grams, and then you will see the analysis where how the COD is being removed in individual stages, and then how bioelectrochemical systems can be incorporated to recover energy. Okay, so anaerobic digester, AD becomes an important process in this case. That is what I would like you to note. And then we have looked at 6 different configurations. So the idea behind this is that we have to enhance energy recovery, right, for the process to be justified. Now, how do we recover energy? So carbon is the form of energy we're looking at. Carbon is the energy currency, okay? So as you will go through these configurations or scenarios, S1, S2, S3, S4, you see the letter C is is changing its size. Sometimes it is small and sometimes it's really big. That is to show that we are manipulating the process, especially the detention time, to increase and enhance carbon capture in the primary clarifier, right? So that can be directed purposely for energy generation processes, that is anaerobic digester in this case. And then we will also see that in the configurations 3, 4, and 6, we have incorporated anaerobic technologies, because as long as we are using a lot of aeration, we are never going to achieve energy neutrality or energy positivity in this process. So if we can introduce anaerobic technologies, and especially now we're talking about Anammox. The reason it's in the red box is that Anammox bacteria is usually pinkish red in color. And so the bacteria, and if we can integrate those to achieve even more, right? Actually, we're removing nitrogen compounds. And so that process is supposed to deliver much higher benefits, energy benefits. So as you see in this figure, I'm showing different zones, red, blue, and green. Green is what we want to achieve. That means we are achieving the higher the green zone, that's the better the configuration is. And then the lesser the red is, the better. And then, of course, the lesser the blue, that is the more the blue is better. We want to push the blue envelope and then shrink the red envelope so that we're able to make the process more green in terms of that's what is blue is the energy recovery or energy potential, and then the red is the consumption. So you will note that S4 and S6 and they are more suited for saving energy, right? Because of the bioelectrochemical system integration. And so I would encourage you to read this paper if you would like to learn more about it. Now, the other way to increase the energy recovery is to introduce external organic resources, right? And then that way, we are utilizing the capacity of anaerobic digester to its full, and we're recovering a lots of methane from there, and that's where we produce more energy. Now, the integrated configurations could be that, you would start with anaerobic digestion and anaerobic processes, as you'll see in this first one, and then you go through the conventional treatment. Only the idea is that it will shrink the conventional treatment process if you still need it. That is the idea. And then here also you see that you can completely go for some certain types of wastewaters. You can completely go towards the anaerobic processes. Okay, and so that is possible, and we'll see that. Next slide will show the real world applications, which we already see. If you're looking in the, resource constrained settings where you do not have a lot of resources, but you have naturally available materials, such as what we talked about, terracotta, biochar, and things like that. So this kind of like septic system where you can directly integrate bioelectrochemical system. And then the other diagram here, it is showing a more, integrated process where you also doing COD removal and nitrogen removal that is allowing for electricity recovery. And then you see that some of the demonstrations, this has been done, and then they have been shown to be working perfectly fine with the separation of urine collected from community and then being utilized in the microbial fuel cells here. And then this is a facility that actually does all of this. So this is happening. Okay, this is not theory anymore. This is not just in laboratory anymore, but this is possible and feasible, but we need to take careful steps in making this process sustainable. Now, environmental sustainability, right? We talk about all of this contributes to environmental sustainability. Now we have carbon sequestration because all of these processes are releasing carbon dioxide. What do we do with this carbon dioxide? Can we again capture them and direct them towards this bioelectrochemical synthesis systems, electrolysis cells, where you can produce more methane and hydrogen and other high value products? Now, so it sounds like everything is going well, right? Everything what I'm showing you, it looked like, yes, this is really exciting and very important and very well motivated for our current context when we talk about circular economy and global environment and sustainability. But what is lacking here? What is the limitation? Why do we not see all of these everywhere? Right? So there is still a lot to be done, although the technologies are known or being explored at the laboratory scale for a long time. We still need to develop a lot more knowledge about how these systems work. There's still knowledge gaps between the microbes and electrode interactions. There is a lot that is behind it. So there is more to understand. Um, how these microorganisms are interacting with the electrode, and then what are the possible ways and what are the preferred ways, and then how they benefit, and then how different microorganisms actually can work together in this microbial biofilm matrix to actually generate that effect. Right. So that is still, there is lots of gaps. And some people propose that synthetic biology interventions could make significant improvements or contributions, but it still needs to be explored. Going to the individual cell level or into the biofilm level and introducing some of these materials to enhance the electron recovery. So it's all about going after these electrons, capturing these electrons. So that is something that needs to be done much deeper level. But my recommendation is always is biological cathodes. There is so much we could do. So cathodic reaction has been highlighted as a rate limiting step because these electrons have to be taken from this loop. Otherwise, there is no flow of electrons. And so cathode needs to be acting fast. Air cathodes are utilized, but they're failing over time except with the use of catalyst. And therefore, now the other way to look at it is aerated biocathodes. You could have certain microorganisms that form films, biofilms, and then aerate them. And so that way you were able to fasten the process, the reduction process at the cathode. But you could also utilize other biocathodes such as microalgae and anomox, which we have shown in our research to be very highly beneficial. That would actually contribute to advanced wastewater treatment. So that's something to be looked into. And then we have lack of experience in pilot scale studies. I'm showing you some of the designs here. When it comes to pilot scale level, we don't have experience, and we also don't have long-term performance experience. And the design aspects are also architectural aspects have to be covered as well. And so these are real pictures where this setup is a modular microbial fuel cell setup where it's being utilized for wastewater treatment. So if we look at laboratory scale studies, there is a lot of ambiguity and discrepancy in terms of the data that is reported, how it is reported in the units. There is no standardization. But anyway, the bottom line is that because everybody is using different materials, different combinations, different volumes, usually the trend is that when you're increasing the volume, the power performance is going down. It's falling down. This is exactly how we show it here. And the data is quite random as well. So it is very difficult to understand where we are, really what is achievable, because everyone is working in different directions there. But because the volume that we need to treat and with the energy loss in the process, it is going to put higher demands for electrode materials and other processes. And so that would increase costs. Now, in future, what we want to do is more long-term pilot scale in situ studies, meaning that we integrate microbial fuel cells in the current wastewater infrastructure. That means on-site demonstration along with or sideline with the actual process to increase productivity, that is including energy generation as well as treatment performance, and then reduce costs. That's what is going to be our goal. So from here, where do we go? With this slide, I'm going to highlight some of the things for our future research directions would be to increase energy recovery, right? That is for electricity for microbial fuel cells, and then high value product recovery, such as methane, hydrogen, and other chemicals in microbial electrosynthesis cells. We need to reduce energy consumption in both of them. We also need to utilize sustainable materials as much as possible. Biochar is the best example for that. But there is so much to do around engineering the biochar to be more suitable, applicable for different applications in this case. Then I strongly suggest utilizing real wastewaters than organic waste or organic waste than synthetic wastewaters. Surprisingly, some of the pilot scale studies are still reporting artificial wastewaters, which is not quite desirable. And then utilizing biological cathodes that could advance functions and then increase circular economy, and then long-term performances, integration with the current technologies. So integrated context will help us to kind of offset some of the drawbacks associated with this technology. And the one thing we need to be mindful about as we develop this technology is to always keep focus on the current and future regulatory context. If we do not work towards these regulations, then we're never going to achieve anything that is meaningful because it's never going to be feasible. And so please keep that in mind as we are kind of curiosity-driven researchers most of the time. We want to do, hey, let me see what happens when you do this. But yeah, that's good. But what is it going to be? What do you expect to happen? And that is going to kind of deliver a more practical result. So that means we need to have a bigger context in our minds, and so that we're able to deliver carefully, deliver more practical solutions. Nexus approach, as I said, food, water, energy nexus, and those kind of topics where we are able to generate multiple products and then enhance the relevance of circular economy. That is highly desirable for this area of research. Now, with that, I'm going to stop here, but I would like to take a moment to advertise some of the things here. I'm looking for graduate research students. If you are interested in any of these things that I do, please do reach out to me. And you also please take a look at my Scholar page on Google. So you will see all different areas that I worked in. And if anything is resonating with your experience, please reach out to me. And also, if you are an undergraduate student in the US, I do have a couple of opportunities for you to work with me directly or to work with my collaborators in Canada. So that is an exciting opportunity that is we're starting this summer with a good stipend and everything covered for you. Please take a look at this as well. And then the research experiences program that we have, in addition to all that we do, these are our summer programs that we do at the Water Institute. If you are interested in working with us, yeah, please take a look at this opportunity as well. And with that, I will stop here and take any questions. Thank you all.